Hello students, in our previous video lecture, we discussed about sexual reproduction flowering plant chapter previous year questions. In this video, I am going to solve most important chapter molecular base of inheritance chapter previous year questions. This is the high weightage and complicated chapter. So now we are going to solve what are all the type of question have been asked from this chapter molecular base of inheritance. Look at the first question. In first question, a DNA fragment has 2500 nucleotides in a DNA fragment, out of which 240 are guanine. 240 guanine. Guanine is a purine. Guanine. G pairs with what? Tell me. G pairs with C with triple hydrogen bond. Right? Am I correct? Now, how many base having double hydrogen bonds between them does this DNA fragment possess? They're asking how many of them are double hydrogen bonds. Double hydrogen bond between A and T. Am I clear? Now, between GC triple hydrogen bond and between A and T is what? Tell me. Double hydrogen bond is there. Am I clear? This is a simple base pair rule. We know this one. What they're asking, the total number of nucleotides are 2500 nucleotides are there. 2500 nucleotides total number, out of which 240 guanine. 240 guanine, number of guanine equal to number of cytosine. We know this one. So number of G equal to number of C. So I can write what? Tell me. 240 cytosine also present. So what is the total number of this G and C here? I can write 480. 480 have triple hydrogen bond between G and C. But in the question, they are asking how many double hydrogen bonds. So 2500 nucleotides, 2500 nucleotide minus 480 nucleotide. If you get 2020, in this 2020 nucleotide, A and T present. So between A, T, what type of hydrogen bonds are there? Double hydrogen bonds are there. So pick up your right answer. They are asking double hydrogen bond. So the right answer is option D is the correct answer. 2020 nucleotides possess double hydrogen bond. Are you with me? You got my point? Come to the next question. Second question. Very, very important question. If there are 999 bases, 999, 999 nucleotides, bases in an RNA that code for a protein. So shall I write it as a mRNA? Right? I'm writing here it is a mRNA. In this mRNA, how many nitrogen bases are there? 999 nitrogen bases are there. We know 3 nucleotide or 3 nitrogenous bases become one codon. Right, one codon. So total 999 nitrogenous base are there divided by 3. So I can write 333 codons. Am I clear? 333 codon. One codon similar or equal to one amino acid. One codon, one amino acid. Look at here with 333 amino acid and base at position 901. Where is the position? 901. 901 is deleted such that the length of RNA become 998 bases. How many codons will be altered? They ask. One nitrogen base got deleted at 901. Fine. Look at here. Total number of nitrogen bases are 999. Total number of nitrogen bases are 999, which is equal to 333 amino acid. That means 333, I can write codon. 333 codon, 333, that will tell me amino acid. Fine. The problem, the deletion of nucleotide at 901. So 901 problem is they deleted here, exactly this portion deleted. What about till 900? Till 900 there is no problem. That means first nucleotide to 900 nucleotide there is no problem. That means first 900 nucleotides are absolutely good. First 900 nucleotide means, shall I write 300 codons and 300 amino acids, correct? 300 codon, 300 amino acid, there is no problem because the actual deletion problem or mutation at 901. So till 900 there is no problem. So 900 nucleotides are good. That means 300 amino acids are, there is no problem. But they're asking, what, tell me, how many codons will be altered, affected? Sir, here 901 problem, 999 till. So that means till here problem is, that means 99 codons. 99 codons will be get affected. 99 nitrogen base, I'm sorry, 99 nitrogen base will get affected. 99 nitrogen base means for how many codons? How many codons, tell me? Divided by 3, that means 33 codons will be get affected. Why 33 codons will be affected? 901 problem is there. 901 problem means we know frame shift mutation, wherever a deletion or addition, from that place to everything will be get altered. Everything will be get altered. So 901 problem to 999, how many are there in between? 99 nitrogenous bases are there. So 99 nitrogenous bases are equal to 33 codons or 33 amino acid. So what is the right answer? How many codons will be get altered? The answer is option B, 33. Total number of nitrogen bases are 999, which is equal to 333 codon, 333 amino acid. Problem 901. So till 900 there is no problem. There is no problem means 300 amino acids are good, 300 codons are good. So problem, where is the problem? 901 to 999 problem is there. That means 99 nitrogen bases. That means 33 codons will get affected. So the right answer of this question, option B, 33. Shall we move on to the next question? Next question. Question number 3, accession reason based question. Accession, primary transcription in eukaryotes are subjected to splicing to remove intron, they mention. We know splicing. Splicing is the process in which removal of intron, introns are non-coding, exons are coding part. So tell me, removal of intron, joining of exons called splicing process. This is an exclusive characteristic feature of eukaryotic transcription. The assertion is absolutely correct. So I'm writing this is the correct one. Right? Assertion is correct. Come to the reason. Primary transcription contains both exon and intron, and the introns are non-functional in eukaryotes. If you are able to recall, from DNA, after transcription process, mRNA will come. But in case of eukaryotes, what happened? From DNA, hnRNA come. What is this hnRNA stands for? Heterogeneous nuclear RNA, which consists intron and exon both. Non-coding, coding both. So this hnRNA undergo a process called splicing. In splicing, what happened? Tell me. Removal of intron, non-coding. It is a waste. It's not going to convert into protein. So what is the importance of intron here? So introns will be removed. So tell me, undergo splicing to form matured mRNA. So in this matured mRNA, only what kind of part is there? Tell me. Exon, coding part only there. And this mRNA directly undergo process called what? Translation to make a protein. Am I clear? So what is the right answer of this question? Both statements are correct and the reason is the correct explanation of assertion. So I'm going with option A is the right answer. Both are correct and it is a correct explanation for the assertion. Next question. Question number 4. A DNA fragment has 3000 nucleotides, same like first question. 3000 nucleotides, out of which 160 are guanine. Here, same thing, 160 guanine. Total number of nucleotides are 3000 nucleotide, out of which 160 is G, guanine, purine, guanine. Number of G equal to number of C. So tell me here, 160 G is there, 160 C also present. We know, just now only I said between G and C, triple hydrogen bonds are there. G. And how many base having double hydrogen bonds? Double hydrogen bond between A and T. So total number of nucleotides are 3000. Now between this G and C, triple hydrogen bond is there. So if you calculate this one, 0 and it will tell me 2 and 1. So 320 is GC. Now I'm writing 3000 minus 320 I'm writing. So the right answer is 2680. 2680 nucleotides are AT. So between them, which bonds are there? Hydrogen. How many bonds are there? Double hydrogen bond. So our answer is option D is the correct answer. In the first question, we have seen 2500 nucleotide, but in this question 3000 nucleotides, same type of, based on same topic. Am I clear? So we are moving on to the next question. Next question is the linking of antibiotic resistant gene. Antibiotic resistant gene. This question from biotechnology principle and process. Cohen and Boyer. First rDNA, antibiotic resistant first rDNA in 1972. Antibiotic resistant gene with the plasmid vector of Salmonella typhimurium by Stanley Cohen and Herbert Boyer was made possible by the enzyme. Simple. If you are able to recall key tools of biotechnology, polymerase enzymes, restriction endonuclease enzyme, vector, ligase enzyme and host. So ligase enzyme like a glue. Ligase enzymes are that enzymes which are used to join the DNA particle. So what is the right answer? The right answer is DNA ligase enzyme. DNA ligase enzyme. Restriction endonuclease enzyme we can cut, but they are asking joining. They are asking gene with plasmid vector of Salmonella typhimurium, linking. That means two DNA fragments are linking. Two DNA fragments are joining with the help of a glue. What is a glue called? DNA ligase. This is also one key tool of rDNA technology. 5 key tools of rDNA technology. So this is the important one which is going to join the two DNA particle to form rDNA. Next question. This is from Principles of Inheritance and Variation. Can you look at this question and answer me what kind of disorder is this based on this pedigree? Based on this pedigree, is this autosomal recessive or autosomal dominant or sex-linked recessive or sex-linked dominant? How can we solve this type of questions? If you closely look at the pedigree, you can get one hint. Parents unaffected, mother, father unaffected, and here offsprings affected, daughter affected, son affected, and son affected. Parents unaffected and offsprings affected is the first general characteristic feature of autosomal recessive. Sir, why can't this is sex-linked recessive? It is autosomal recessive, I said. So why can't it is a sex-linked recessive? We'll see. Let's take mother is a carrier, XCX, color blindness, XCX, mother is a carrier, father is normal, X and Y. X and Y. Okay, we can understand this boy is affected because of mother X chromosome XC and father Y chromosome. Okay, I understand and I can what they call I understand. I agree with this one. This boy is affected. Fine. This boy also affected. I can agree. XC and also Y is the genotype. How come this girl affect? Tell me. How come this girl affect? If this girl is affecting, that means father or mother, any one of these two would have affected. But in the question, both parents are normal. Both parents are normal. How can this girl affected? So this is not sex-linked recessive. This is not sex-linked recessive. So what is this? This is autosomal. So I can write, tell me, both parents are unaffected. So shall I write here capital A, small a, capital A, small a. This boy is small a, small a, small a, small a, small a, small a. Why small a, small a? When both genes are recessive condition, the disease will be expressed. Sickle cell anemia, thalassemia, cystic fibrosis, phenylketonuria. These all are autosomal recessive. Albinism also autosomal recessive. So I can go option C is the correct answer. I hope you all are following me. Next question, question number 7. Which was the last of 24 human chromosome to be completely sequenced? If you remember Human Genome Project, HGP, HGP, human genome process. Genome means haploid number of chromosomes. So human haploid number of chromosomes are 23 only, but in the question they mentioned 24. How come 24? Listen carefully. In the last class, in the theory class, we discussed this one. 22 autosomes, right? And tell me, 2 sex chromosome, X chromosome and Y chromosome, both chromosomes. So 22, 23 and 24. So total in human genome project, total how many number of chromosomes they sequenced? 24 number of chromosomes they sequenced. What kind of methodology they use? Sanger's method. HGP, human genome project, 24 total chromosomes. You know, the last chromosome was chromosome number 1. If you able to recall the salient features of HGP, human genome project, I think if I am not wrong, the second last point, chromosome number 1 has large number of genes. Chromosome number 1, this chromosome number 1 consists 2968 genes are there, along with one largest gene. If you are able to recall the human genome project salient features, in that one it is clearly mentioned average gene consists. Average gene consists. Tell me, 3000 bases. 3000 bases. But the largest gene name is what? Dystrophin. Dystrophin gene is located on chromosome number 1. This is the largest gene. This largest gene, you know how many nucleotides given bases mentioned? 2.4 million. 2.4 million bases. Remember all of you. So largest gene and also large number of genes, also chromosome number 1. So the scientists took little more here, and this is the last chromosome because it took little more time to sequence. That's why 1990 HGP started. In the year of 1990, it was actually 13-year program. 13 years program. It should have been completed in 2003, but it was completed in 2006. You know why 3 more years extra? For chromosome number 1 sequence, because it is the largest chromosome. Large number of genes are there. And also what, tell me, the largest gene also located on chromosome number 1, that is dystrophin gene. So the right answer of this question is option A is the right answer. Am I clear, students? Shall we move on to the next question? Question number 8. Question number 8. Which of the option has correct identification PQR in the illustration of central dogma given below? One central dogma given, here it is given. Very simple. Any student can answer this one. Any students. From DNA to DNA, it is called a DNA replication. And from DNA to mRNA, what is this P? This P is called what? Transcription. This process called transcription. Transcription. Next, here Q is what, tell me? Directly I'm writing mRNA. And mRNA undergo process to form a protein. What is this process called? Translation. Translation process takes place in cytoplasm. Replication and transcription takes place in where? Nucleus. So find out the correct answer. P is transcription. P is transcription, Q is mRNA, R is translation. So option D is the correct answer. Simplest question. Central dogma diagrammatic question, simplest one. Tell me here, I'll ask one question here. DNA to DNA, DNA polymerase enzyme. DNA to RNA, RNA polymerase enzyme. Am I clear? RNA polymerase enzyme for the transcription. Done. Next question. Next question. In a 68 nanometer long of DNA, adenine constitute 25%. Calculate the total number of hydrogen bonds in the DNA. How can we solve this type of analytical, little higher order thinking questions? This is a most important type of question. This is a very important question. Carefully look at here what I'm explaining. 68 nanometer long of DNA they mention. 68 nanometer long of DNA. 68 nanometer divided by, I am writing 3.4 nanometer. 3.4 nanometer is what? 1 ton of DNA is 3.4 nanometer. 3.4 nanometer is 1 ton. Yes or no? How come it is 3.5 nanometer? The distance between the two consecutive base pair is how much? 0.34 into 10 base pairs are there. I told you 10 base pairs in 1 ton. So tell me, it will come to 3.4 nanometer in, tell me, 1 ton of DNA. 1 ton of DNA, right? In 1 ton of, what is the length of 1 ton of DNA? 3.4 nanometer. How many nanometer is given? 68 nanometer. Shall I write, if I calculate about 20 tons? 20 tons. 68 nanometer divided by 3.4 nanometer, it is 20 tons. In each turn, 10 base pair. Yes or no? So in each turn, 1 turn consists, what, tell me? 10 base pairs, right? Now here, how many turns are there? 20 turns. So 20 into 10 base pairs. If you write like this, you will get, tell me, total 200 base pairs. It is a base pair. Two nucleotides are there. So total number of nucleotides, how many? Into, I can write 2. So 400 nucleotides or 400 bases. Am I clear? 400 bases. Now you look at that one. 25% of adenine is given. So out of 400, 25 out of 400, 25% is adenine. So what is the 25% of 400? Tell me. 100. So 100 adenine. So 100 thymine. A equal to T, right? A equal to T. 25% is A, means 25% T also there. So between them, how many hydrogen bonds are there? Double hydrogen bonds are there. A and T, double hydrogen bonds are there. So 100 into 2, so total 200 hydrogen bonds. Total 200 hydrogen bonds. Now here 25% A, 25% T. Shall I write 25% C also? So tell me here, out of 400 nucleotide, 25% C means what? Tell me. 100 C, 100 G, because C also 25% and G also 25%. Am I clear? G also 25%. So 25 to total 100% DNA we got. Am I clear? Now between G and C, triple hydrogen bonds are there. Triple hydrogen bonds are there. So I'm writing 100 into 3. Why I'm writing 3 here? Triple hydrogen bonds, right? Triple hydrogen bond between G and C. So total here it is 300. Now here it is 200. So total number of hydrogen bonds in the DNA 500. Very easy. If you know little concept, if you are perfect in concept, you can easily understand this type of questions. One more time I'm explaining. Carefully listen. 68 nanometer long of DNA they mention. 68 nanometer long. We don't know how many nucleotides are there. We don't know. Just they give it 68 nanometer long. First, we need to calculate the total number of nucleotide. If you calculate the total number of nucleotide, automatically here adenine A 25%. What is the number of adenine? We can easily get. So, in order to do that one, 68 nanometer long DNA divided by 3.4. 3.4 is what? 1 ton of DNA. 1 ton length. 1 ton length is 3.4. So I'm getting 20 tons. I know 1 ton 10 base pair. 20 tons are there. 20 into 10. So total 200 base pairs I got. 1 base pair equal to 2 nucleotide. So total 200 base pair into 2, 400 nucleotides are there. I got. So total number of nucleotides. Now we solve. Easily now 25% of adenine in 400. So tell me 400 nucleotide, 25 adenine means 100 will come. We know A equal to T. So A 100, T also 100. Between A, T, double hydrogen bonds are there. So we got total number of hydrogen bond between A, T only we got. C, G also left. Read the question. In the question they are asking, calculate the number of hydrogen bonds in that DNA. They are asking the total number of hydrogen bonds we need to find out in the DNA. Just now we got A, T only we got. C, G also there. So C how many? 25. That means 100. G how many? 25, that means 100. Between CG, triple hydrogen bonds are there. So 300 plus this 200. So total 500. So the right answer is 500 hydrogen bonds. Am I clear? Shall we move on to the next question? Next question. A DNA replication for is shown below. Okay, this is a DNA replication for. Identify the option with correct polarity of the strand. Very easy. Diagrammatic questions. We know new DNA always polymerize from which prime to which prime? Tell me. 5' towards the 3'. Right, 5' toward the 3'. I can call this A 3' and B is 5'. Shall I call it as a continuous or leading strand? Continuous or leading strand. So E is what? Tell me. It is opposite, and opposite, what they call anti-parallel. So 5', and F I can write it is a 3', right? So what about the opposite of this one? A, C. Here it is A 3' to 5'. This must be 5' to 3'. 5' to 3'. Where is a new DNA? I told you new DNA only polymerase from 5' to 3'. So I can write this is here, it is 5' to 3'. So find out the correct answer. AB is what? Tell me. 3' to 5'. 3' to 5'. Right? Next, EF. This is EF. E 5', F. So 5' and 3'. This is appropriate correct answer. Come to the B, C, D also will check. B, A, B is 5' to 3'. A, B, no, it is not 5' to 3'. Leading strand is from where to where? 3' to 5'. Parental template strand, right? Now, CD is mentioned. Where is CD? CD is what? This C and D. CD is what? 3 to 5 they mentioned. No, it is not 3 to 5. CD, 5 to 3 they mentioned. Okay, it is 5 to 3. I agree with this one. What about EF? EF is 3 to 5 mentioned. No, it is not 3 to 5, it is 5 to 3. Remember, new DNA always polymerized from 5' to 3'. DNA dependent DNA polymerase enzyme only polymerize the DNA from 5' towards the 3'. So look at here, 5' toward the 3', 5' toward the 3'. Am I clear? This is called what? Okazaki fragment or discontinuous strand or lagging strand. Correct, student? Come to the next question. So the right answer of this question is what? Option A is the right answer. Am I clear, students? Come to the next one. Very, very, very easiest question. Look at here. There are 2 pentose sugars given. 2 pentose sugar, pentose, 5 carbon compound. Biomolecule chapter in the 11th grade also we discussed. Pentose sugars are 2 types. Ribose, deoxyribose. Done. Look at here. This is carbon number 1, carbon number 2, carbon number 3, carbon number 4, and carbon number 5. 5 carbon compound. Carbon number 2, carbon number 3, OH groups are present. We call what? This is a ribose sugar. And this ribose sugar present in where? RNA. Look at here, all 1 oxygen less, 3rd carbon only OH is present. Deoxyribose sugar. This deoxyribose sugar present in where? DNA. Out of these 2, which one is more reactive? RNA more reactive, less stable. DNA more stable, less reactive. Find out the correct answer. Find out the correct answer, students. A is ribose, correct, ribose, which is quite stable. It is less stable. Now, A is deoxyribose. No, A is not deoxyribose. A is ribose, correct, which is reactive. Absolutely. Next, B is deoxy, that is reactive. It is constant. So the right answer is what? Option C is the correct answer. Simple question. One is pentose, ribose sugar present in RNA. One is deoxyribose present in the DNA. Clear? Next, come to the next question. Purines found both in DNA and RNA are mentioned. Purines, RNA and DNA. What are purines? Nitrogen bases are 2 types, right? Nitrogen base 2 types. One is called purine. Second one is called pyrimidine. Yes or no? Pyrimidine. Purine and pyrimidine. Purines are A and G. Pyrimidines are C, T and U. Cytosine, thymine and uracil. Cytosine, thymine and uracil. Now A, G, C, T present in where? DNA, right? And A, G, C, U present in where? RNA. The question asking, purine found both DNA, RNA. Both DNA, RNA. Purines are AG. Both are present in DNA, RNA. Find out the correct answer. Cytosine and thymine, adenine and thymine, adenine and guanine. So the right answer is option C, purines. AG, purines. Just now I said A plus G by C plus T equal to 1. Who is this? Who proposed this one? This is Chargaff rule. Number of purines equal to number of pyrimidine. So number of purines equal to number of pyrimidine, and also I clearly mentioned in the previous one, A pairs with T with double hydrogen bond, G pairs with C with triple hydrogen bond. So adenine and guanine common in DNA and RNA. So where is the difference? Now T present in the DNA, but here RNA U present, T absent. So the difference in the pyrimidines, not in the purine. So the right answer is what? Adenine and guanine. Next question. Reverse transcriptase. Reverse transcriptase. What is the meaning of reverse transcriptase? DNA to RNA formation called transcription. Transcription, we know this one, simple. And RNA to formation of protein known as translation. Am I clear? Translation. Fine, student. This is a DNA genetic material organisms. Central dogma. What about the RNA like HIV viruses? In HIV viruses, first RNA will make DNA. This process is called what? Tell me. Reverse transcription. Reverse transcription. What is the enzyme name? RNA dependent DNA polymerase. RNA dependent DNA polymerase. They're depending on RNA to form DNA. Such DNA called cDNA, complementary DNA. And from this DNA, again RNA form. This is called transcription, normal transcription. And from here to protein formation takes place called translation. This is actually RNA genetic material organism. Find out the correct answer. Find out the correct answer. Reverse transcriptase is DNA dependent DNA? No. DNA dependent RNA? No. RNA dependent RNA? No. RNA dependent DNA polymerase. Option D. Here it is RNA polymerase enzyme. What will form? It will form mRNA, rRNA, and also tRNA. It will form RNA polymerase. Am I clear? Comes the next question. Question number 14. The type of bond represented by dotted, this dot in a schematic nucleotide, polynucleotide chain. If you know the structure of nucleotide, let me draw one. This is pentose sugar. I'm taking DNA nucleotide. Okay, carbon number 1. This carbon number 1, adenine. That means nucleotide attached. First carbon of pentose sugar, nitrogen base attached. N-glycosidic bond. The bond name is N-glycosidic bond. Carbon number 2, carbon number 3. Carbon number 2, H, H. Carbon number 3, H, OH. Carbon number 4, H. Carbon number 5, CH2OH. This is nucleoside. Because 2 compounds only, pentose sugar, and also what? Nitrogen base is there. So what exactly this bond name? N-glycosidic bond. So what is this dot? This dot is actually, B stands for what? Nitrogen base. S stands for what? Sugar. P stands for what? Phosphoric acid. That was a phosphodiester bond, 5th carbon. Here it is what? Tell me. P double bond O, OH, right? Here it is OH and also OH. This CH2 I'm writing here. This CH2OH I'm writing like this. Shall I write like this? OH, CH2. By the loss of water molecule, phosphoric acid attached to the carbon number 5. Which bond? Phosphodiester bond. But they're asking nitrogen base to sugar. This is N-glycosidic linkage. It is not hydrogen bond. Hydrogen bond between one nucleotide to other nucleotide. Two different DNA strands. Hydrogen bonds are there. Two different DNA strands, two complementary DNA strands between, there is a hydrogen bond is there. But between nucleotide, let me take here A, G, C, T, A. So between two nucleotide of same DNA strand, phosphodiester bond is there. But when you write the complementary C, triple, G, triple, A, double, T, double. This is 5 dash. Opposite and anti-parallel 3 prime. Here it is 3 prime and this is 5 prime. So what type of bond between the pentose sugar and nitrogen base? N-glycosidic bond. Am I clear? Come to the next question. A person with trisomy 21. Trisomy 21 is what? Turner, Down, Klinefelter. Can you guess? Can you guess 21st trisomy? So 21st trisomy is a Down syndrome, autosomal trisomy. 21st autosomal trisomy. Now, it is furrowed tongue. Yes, correct. What they call characteristic palms is right, correct. Rudimentary ovary, no. Rudimentary ovary, Turner syndrome, monosomy, X0. But this is a trisomy. This is wrong. Third one is wrong. Gynecomastia. Gynecomastia in Klinefelter syndrome, XXY. So this also not correct. So the right answer is 1 and 2. Where is 1 and 2? 1 and 2. Option D is the correct answer. It is aneuploidy trisomy, Down syndrome, autosomal trisomy. This is male and female, it can see. But gynecomastia, Klinefelter syndrome in males and Turner syndrome, rudimentary ovaries in female. Remember, let me write here XO. Here it is XXY. Keep in mind. Shall we move on to the next question? Next question is 16th question. Match the following with the lac operon listed in A and their respected column with B. They are given I gene, Z gene, A gene, Y gene, structural genes. I gene code for what? Repressor protein. Very easy. Z gene code for what? Beta galactosidase enzyme. A gene code for what? A gene code for trans-acetylase enzyme. Y gene code for what? Permease enzyme. So 3, first one is 3, next B is 1 and C is 4 and Y is what they call 2. 3, 1, 4, 2. Find out the 3, 1, 4, 2. 3, 1, 4 and 2. Option C is the correct answer. Lac operon question. Easy question. I'm not much explaining about this one. Shall we move on to the next one? Next question. If both the parents are carrier for thalassemia, what is thalassemia? Sex-linked or autosomal? So thalassemia is qualitative or quantitative? It is a quantitative blood bleeding disorder of autosomal recessive. Quality, quantity. Quantity is thalassemia, quality is sickle cell anemia. So here thalassemia, both parents are carrier. So capital A cross with capital A, both parents are carrier. They are not showing disease. Both parents are carrier. And the chance of the affected child, they are asking chance of the affected child, capital A, small a, capital A, small a. So what we will get? Capital A, capital A, capital A, small a, capital A, small a, small a, small a. 25% chance is there. Find out the correct answer. The right answer is option A. These three are what? Tell me unaffected, because there is a dominant gene is there. I told you recessive condition only will express when two recessive genes present. Look at the two recessive genes. So 25%. Shall we move on to the next question? Next question. Look at here. If the sequence of nitrogen base, of the coding strand is given 5 dash. Coding strand is what? 5 dash. Look at here. A, T, G, A, T, G, and this is 3 prime, right? In a transcription unit is 5 dash to 3 dash. The sequence of RNA transcription they given. Easy, very, very, very easy. We know coding and mRNA same except T and U. except T and U. So I'm writing what? Tell me, mRNA 5 dash, same, right, except T plus U. A, U, G, A, U, G, and 3 prime. This is mRNA. This is mRNA. You no need to write the complementary and you no need to make a mRNA. Very easy. Find out the correct answer now. 5 dash to 3 dash. 5 dash to AUG, AUG, AAU, AAU, G, and 3 prime. Answer is option A. How simple? How simple, very easy. Coding stand mRNA same except TU. Except, okay, if they mention template. If they mention template, look at here. If they mention here 3 prime, if they mention like this 3 dash, okay. Here it is, what? Tell me, T, A, C, and T, T, A, C, and this is 5 dash. They're like this template stand they given. And they're asking you to write mRNA. And they're asking you to write a mRNA. This time don't write same except T plus U. That will be wrong. You need to make a direct complementary here. So right here 5 prime, T pairs with A pairs with T, but you're writing RNA, right? T is not there. Place of T, what is there? U is there. So I'm writing U, C plus G, A plus T plus A, T plus A. Next here it is what? Tell me. Here it is U and G, and this is 3 prime. Look at here. This one and this mRNA same. AUG, AUG, AAU, AUG. Am I correct? Am I clear? Shall we move on to the next question? Next question. 1 turn of the helix of B form DNA, approximately 1 turn. Now you answer me. I explained you this one in the previous questions. 1 turn DNA. Sir, if it is a base, what they call consecutive base pair, 0.34. If it is a 1 turn, simple answer is what? 3.4 nanometer. 1 turn, 3.4 nanometer. If it is a consecutive base pair, 0.34. Simple. Done. Next question. Question number 20. Very, very important. This academic year, this question, there is a high chance to come. RNA polymerase enzyme. RNA polymerase enzyme. My dear students, keep in mind and write down in your notes. DNA to formation of RNA. DNA to formation of RNA we call what? Transcription. Transcription. But here there are 3 different type of RNAs are required for the synthesis of protein. 3 different type of RNA. What are the 3 different type of RNA? One is called rRNA, one is called mRNA or hnRNA, next tRNA. For the synthesis of this 3 different type of RNA, we need 3 different type of RNA polymerase enzymes. So RNA polymerase enzyme, RNA polymerase enzymes are 3 types. What are the 3 types? Tell me. RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. 3 different type of RNA polymerase enzymes. RNA polymerase 1 is important for rRNA transcription. RNA polymerase 2 for mRNA or hnRNA. mRNA or hnRNA. RNA polymerase 3 for tRNA. Find out the correct answer now. Carefully read this one. You will. This is only in eukaryotes. Don't get confused in the exam. If they're asking bacterial transcription, which of the following enzyme required? Only 1 type of enzyme in bacteria. I'm talking about eukaryotes. This is eukaryotes. Look at the question. Match the column RNA polymerase with the transcribed product. Here it is 1, 2, 3 mentioned. That means they're talking about the eukaryote transcription only. So look at here. RNA polymerase 1, rRNA. RNA polymerase 2, hnRNA or mRNA. I'm writing here hnRNA or mRNA. RNA polymerase 3, tRNA. RNA polymerase 3, tRNA. So tell me 2, 3, 1. Where is 2, 3, 1? 2, 3, 1. Option C is the correct answer. 2, 3, 1, right? 2, 3, 1. 2, 3, 1. But in the exam, if question come, How many RNA polymerase enzyme required for the synthesis of RNA, rRNA, tRNA, mRNA in bacteria or in prokaryote? Our answer is one only. Single RNA polymerase enzyme is enough for the transcription of any type of RNA in bacteria. But in eukaryote, it's not like that. It is separate. 1, 2, 3, separate. Clear? Come to the next question. 21st question. AGGTATCGCA is a sequence from coding strand of a gene. What will be the corresponding sequence of transcribed mRNA? Coding strand from 5' to 3'. So directly they give a coding strand. So right here everything same except T plus U. Now right here A G G U A U C G C A U. Find out the correct answer now. Where is the right answer? A G G U A U A G G U A U C G C A U. Right answer option A is the correct answer. They are given a coding strand. Coding strand always from where? 5' towards the 3'. So mRNA also from where? 5' towards the 3'. This is what? mRNA. I told you coding and mRNA almost similar. T plus only U. You understand my point? Come to the next question. Name the enzyme that facilitate opening of DNA helix during transcription. Opening DNA helix. I know you all are thinking DNA helicase enzyme. But read the question carefully. They are not asking opening DNA helix during replication. They are not asking. They are asking transcription. So transcription, RNA formation. RNA formation means the enzyme name is what? Tell me. RNA polymerase. It is not helicase enzyme. I know whenever you read this opening of helix of double-stranded DNA, you will go with the helicase enzyme. No, they are not asking in a replication. They are asking in a transcription. So when they are asking in a transcription, our answer is RNA polymerase. What about DNA ligase? I told you 2 DNA particle joining for this process, DNA ligase enzyme. But here it is not joining. Leave it. DNA helicase enzyme can cut the hydrogen bonds and open the DNA helicase during replication, not transcription. Now, DNA polymerase enzyme can polymerize the DNA to DNA. This process called what? DNA replication. But in our question, they asked transcription. So, transcription, RNA polymerase. Am I clear, students? Are you following me? Come to the next question. Which of the following rRNA act as? rRNA, ribosomal RNA, act as a structural RNA as well as ribozyme in bacteria. Ribozyme, non-proteinaceous enzyme. This is behave like an enzyme, but it is not a protein. Formation of peptide bond. This enzyme is important for the formation of peptide bond during translation process in the large subunit of what they call ribosome. So 23S rRNA. 23S rRNA is what? Tell me. rRNA act as a structural what they call ribozyme, direct encapsulation line. So option C is the correct answer. Next question. Types of RNA polymerase require in nucleus of it is not nucleus, it is in of prokaryotes of RNA synthesis they mentioned. Prokaryotes. How many different type of RNA? How many different type of RNA polymerase in prokaryotes? What is your answer? I told you this one. If it is a eukaryotic transcription, we would have go with the 3 RNA polymerase 1, 2, 3. But they're asking prokaryotes. What is the answer? The right answer is only one single RNA polymerase enzyme enough to transcribe rRNA, mRNA, and also tRNA in case of prokaryotes. Am I clear, students? So these are the 24 questions PYQ, and few questions from NEET PYQ also there, and few questions from CBSE PYQ also there. Most important chapter. One more time you watch this video and practice more MCQ question. You should not lose a single mark from this chapter. 95 plus and 100 out of 100 is our target in this academic year 2025. Biology 100 out of 100. So higher weightage chapter, high weightage chapters you should not ignore. Keep on practicing more MCQs. Thank you so much.